That's that. So just wanted to start with some case series, and I initially thought this was more CLL focused one, so I actually picked two CLL case series, but I think in theory can apply to other lymphomas too. But just to highlight the patients that I'm kind of typically seeing in clinic for CAR T cells now, in the era of multiple therapies that we have seen, is that the first one is a 65-year-old guy with CLL, unmutated, the deletion 11q, diagnosed a long time ago, and then got first-line therapy in FCR. Good response initially, but then relapse. Got another trial with an idelalisib plus rituximab. Partial response, but stable disease, but then relapse again. Got on ibrutinib. Again, good response initially, but then relapse again, and then got started into venetoclax. But stable disease now, but persistent. So what are the possible next steps in treatment who had multiple therapies in this patient before? Should we be more proactive and get another therapy, or what are the other treatment options other than clinical trial, and what clinical trials may be appropriate for them? The second is kind of young patient that I saw in clinic, and also CLL with deletion 17p with a complex karyotype, initially diagnosed, and then started treatment a couple of years later after diagnosis with progressive disease, and then with ibrutinib, and she achieved a PR. But still with a persistent disease, about 20% disease in the bone marrow biopsy, and then the palpable lymphadenopathy. And she was referred to transplant doctor because of complex karyotype deletion 17p. She did have a donor, but then also coming to the clinic for can there be other options other than transplant. So these are kind of the probably typical population that I'm seeing in clinic right now. So in terms of the CAR design, so what is CAR? I think the first part might have covered a little bit. You guys heard a lot about them. So CAR really refers to, it stands for chimeric antigen receptors. It's an artificial T-cell receptor molecule. So it's a combination of you take the monoclonal antibody binding domain, there's a single chain variable domain. I'm trying to see, there's a pointer here. Oh, there you go. This is the pointer, right. Right. So here is the binding domain, the single chain variable domain, that you connect them to the T-cell receptor, the signaling moiety here. So this is a transmembrane domain. This is actually a signaling molecule. Predominantly CD3 zeta chain is the most common used in clinical trials now too. So it's a combination of the antibody and then the T-cell receptor. So it allows to bind like an antibody without in the context of HLA or MHC regulation. So there's universal applicability in that sense, but it acts like T cells. And then I think previous speaker talked about first generation, second generation. So all the clinical trials that I'll be talking about in this talk will be using second generation CAR. There's some preclinical and clinical evidence of third generation that combine two co-stim domain, either with the CD2841BB. These are common limited. So when you actually combine them together, thinking they're going to be more potent, these cells actually don't expand very well. They get exhausted very quick and they die out. So these haven't been developed really further in that sense. So there's really the second generation CAR, which the difference between first and second is containing the costimulatory domain. So either CD28 and 41BB, those are the two predominant co-stim domain that's in clinical trials right now. So how does it work as a cancer therapy? So there are a couple different ways you can do it, but most common way is autologous T cells, meaning or the patient-derived T cells. The patient will first of all undergo leukapheresis or T cell collection process, and then you take the T cell product or the white blood cell product and isolate the T cells, and then you stimulate those T cells and using either retrovirus or the lentivirus. The viral derived method is one of the ways, but there's also non-viral the transduction method using Sleeping Beauty or transposons. But the idea is that you insert the CAR gene, the chimeric antigen receptor gene, into the native T cells that you have collected. You then expand these T cells for the period of the time, the average about 7 to 14 days, depending on the mode of method and then the stimulation transduction. So you grow these T cells out. And then most common way is that you can actually cryopreserve them, which allows the flexibility of the T cells, or you can infuse them fresh too. So the whole process, in theory, in ideal situation, will take about 2 weeks. But in reality, they're getting into clinical trials and things like that will take more time. So that's actually the reality kind of kicks in. But these are the basic concept of what CAR T cells will look like. And during the T cell production time, depending on how long it takes and depending on the clinical trial availability and then slots, then some of these patients will likely require some interim therapy before they receive the CAR T cells. So what are the advantages of the CAR T-cell therapy? As we mentioned before, because it's utilizing the monoclonal binding domain, so it's HLA-independent antigen recognition and therefore universal application in this sense. So any target antigen that's in the surface is a targetable target. They include proteins, carbohydrates, and glycolipids. And you can actually generate these tumor-specific T cells in relatively short period of time. It's getting shorter and shorter. On average, it still takes about 7 to 10 days on average. Because you're using patient's own T cells, there's a minimal risk of graft-versus-host disease. Although if the patient had a prior transplant and you're collecting the T cells from that patient, there's a mixed chimerism there too. But all the clinical trials that's ongoing right now, actually I shouldn't say all, most of the clinical trials are including patients who don't have active graft-versus-host disease. But there's also clinical trials for ALL, the patients who had a donor-derived T cells too. So there's actually those situations, there may be potentially a little bit more risk of graft-versus-host disease, although clinically they have not been seen yet. So one advantage is that this is a potential for living drug. There's kind of the constant, there's a lot of room for improvement there too, which we'll kind of talk about. So ideally, there's T cells as opposed to antibody, because these T cells can self-replicate and generate, that there's potential for long-lasting immunity. So there's active immunization kind of the process. So select modification specific T cell subtypes. So because it's just a gene modification, you can select a particular T cell subtypes you want to modify, CD4, CD8 in a particular ratio, in a particular T cell subtypes. And also for that matter, for the second generation, you saw the kind of evolution of the CAR. You can actually evolve even further, which we'll talk about at the later part of this talk, to see what the future will look like. So there's kind of the possibility for additional modification. So the most important thing for these CAR T cells to work very well and safely is a target selection. So we talk about what kind of things could be target. Well, you basically want to minimize on-target off-tumor toxicity, and there have been some previous incidents that this could be problematic. So if your target, if the CAR is targeting an antigen that's also expressed in normal tissues, obviously, then it can be potentially detrimental for this or fatal for these patients, which has happened previously. So that's really the first step in choosing the appropriate target for the CAR T cells. Sometimes it's unpredictable. Some of the TCR or T cell receptor enhanced the therapy, cellular therapy, no matter how well you screen them, there's always a potential to cross-reactivity for those. So there's a lot of testing that now needs to be done in vitro before moving this type of therapy to clinical trials to prove as best as you can, although not perfect, that there will be a very little cross-reactivity that you expect. And also need to consider potential impact of long-term suppression. So if your CAR T cells work very well, or they persist very long, and they're targeting in normal tissues or normal cells, then you need to really kind of think about what are the consequences can we afford that? So I'll be talking about the CD19, which is probably it is the most advanced in the setting of the CAR T cell trial as a targeted therapy. So CD19 is an attractive target because it's expressed in various maturation stages of the B cells, starting from the pre-B cell ALL, pro-B cells and pre-B cells that's mutated in pre-B ALL, and all the way to mature B cells for CLS and mature lymphoma cells, and a very small subset of the plasma cells will express CD19. And a CD22 and a CD20, and there are CARs for that too. The CD22 is a good target for its monoclonal antibodies, immunoconjugate, and the CARs being developed in clinical trials, but these are a little bit earlier stages of the clinical development, so most of the data that we're talking about will be using CD19. So I talk about, but not all CARs is the same. So we talk about second generation CARs in the clinical trials. I think it's important to recognize that these are kind of somewhat complicated process. So these are just some of the examples of this CAR in clinical trials that's ongoing right now for mostly in the CLL, non-Hodgkin lymphoma, and in ALL patients. But so there are several places they can differ in the vectors or the CAR design. So where you're deriving the monoclonal, the single chain variable domain, so they could be derived from different hybridoma, and therefore they could have a different antigen-based binding affinity that may matter. And then the signaling domain, we talked about the co-stim CD28 versus 41BB. So they are kind of the largely two different vector designs there too, whether that matters or not. We can talk about those, but these are some of the things to think about. And another very important factor that we begin to realize now is that these vectors are one problem, but how you manufacture the cells is another issue too, because we talked about how you stimulate the T cells and how you modify those T cells. We used to think they may not matter as much, but I think the more data to show that during the process, even though you're using from the same backbones, you can actually change the phenotype of the cells during the process too, and therefore you can make them more active, and also the toxicity profile may differ too. So I think there are a lot of factors that go into which makes it very challenging to compare product to product or generalize that one product is equal. So I think there's a lot of kind of the process that we have to think about when we kind of interpret the data in the setting of the toxicity and the efficacy as well. So what are the basic scheme of the CAR T-cell trials too? I think we saw the diagram. So basically the patients that will get enrolled, these are all experimental therapy investigation agents. So it will be in the setting of the clinical trials, the patients that once deemed to be eligible, they'll go under leukapheresis with a T-cell collection. The T-cell production is the part that I showed you before, and during the process, most of the patients will likely receive some salvage chemotherapy. And then after that, there is a part of the conditioning chemotherapy, some will call lymphodepleting chemotherapy. The purpose of this chemo is really not to tumor reduce, cytoreduce, although they may happen depending on which chemotherapy that you may choose. The predominant mode, the reason that we do this is that because it allows the T cells to expand further. So the previous clinical trials which we have done back in 2006 and 2007, without conditioning chemotherapy, we realized that these T cells don't expand very well, and then therefore doesn't really work. So we went back to the lab and modified the trials, and as well as others have done. The conditioning chemotherapy, at least with the second generation CAR that we're using now, is a kind of the essential component of these CAR T cells to work very well. But there's a variable component of the conditioning chemotherapy. But this is a basic scheme of what the T-cell trial will look like. But there are some differences in all the trials that we'll be highlighting. The CAR T-cell component that we talked about, the mode of transduction, we kind of talked about that there are different ways to modify the T-cells too. How you manufacture the cells and cell manufacturing, what kind of stuff you're adding to the process is also different. Cell source, this patient-derived versus donor-derived, and then the patient population, adults versus pediatrics, patient had prior transplant or not, and then the method of lymphodepletion to meaning the conditioning chemotherapy. There are really various chemotherapy regimens, and because of that, they may also impact the toxicity or maybe the efficacy of the CAR T cells as well. So the CAR T cells, using CD19 CAR T cells, have first shown to be efficacious in the various groups in the United States in 2010 and 2011. First in indolent B-cell lymphomas in the CLL. This is the data from the NCI showing the follicular lymphoma patients, and this is the data from the UPenn in the CLL patients, and this was the data from us, our group at MSK, showing in the CLL patients. So there have been some evidence from the 2011 on that these T cells actually work very well. So this is a T-cell the CAR T-cell data from the UPenn. This is published by Dr. David Porter in the CLL patients. So their product is called CTL019. So this is actually 19 targeted with a 41BB as a co-stim domain using lentiviral transduction. So this is a patient population they published last year, and the 14 patients, the patient with a CLL with the median previous therapies of the five previous therapies there. These are high-risk patients, likely about 50% of the patients had deletion 17p, but heavily pretreated patients. Their clinical trials allowed a kind of different lymphodepleting chemotherapy. So some received the bendamustine, some received fludarabine and cyclophosphamide, and some also received pentostatin and cyclophosphamide. I should mention Cytox and cyclophosphamide is probably the most common conditioning chemotherapy that we and others have used, but also there are some evidence that adding fludarabine may enhance the T-cell expansion and persistence. So we will see a lot of data with the flu-cyc combination. But as you can see, there are other kind of particular conditioning chemotherapy that could be combined with its CAR T cells as well. So this is the summary slide from their study of the response and follow-up. They're in this the 14 patients, they saw 4 CRs, which kind of give you about 29%, and the PR 29%, and there are kind of a good number of the non-responders too. So CR rate is actually lower than what we expect in ALL patients in general. We actually thought that this will actually work much better in a CLL and non-Hodgkin lymphoma than in ALL. So we actually, the first clinical trials are actually entering in CLL and the subsequent move to ALL, and then ALL, it worked very well. It's about 80 to 90% CR rate. This is all where the most approval or the advanced clinical trials are happening right now. So this was somewhat surprising when we saw the CLL data is was lower, although now we begin to understand why that may be the case, which we'll kind of talk about. But having said that, 30% CR rate in this heavily pre-treated patient population is very meaningful too. What's important about this study was that all CR patients remained in CR in long-term follow-up. So even just kind of similar to previous data, that not a lot of patients may benefit, but once you get the benefit, those patients have a very durable outcome. So this is the overall survival and progression-free survival from the data from the same study as well. This is showing the T-cell persistence in vivo. So this is just to highlight that there are differences in terms of the pharmacokinetics or the T-cell expansion and persistence in the patients who are good responders, complete response, PR, and the non-responders too. So non-responders, as kind of expectedly, they have a very little T-cell expansions or very short-term T-cell persistence, as opposed to CAR T-cell patients kind of going, this is the first year all the way up to the year. A lot of these patients, actually all of these patients had a T-cell detectable at the time of 1 year mark too. And this is going beyond 1 year, some of the patients up to 4 years, using 2 different ways of detecting T cells, either by flow or PCR. They are able to detect some of the T cells in a long term, in CR patients only. So showing the T cell persistence, at least in this data set using 4-1BB, that it appears to be correlating with the good response and durable response. So, and then this was also mentioned before too, the CTL with the same study and the DLBCL patients, Dr. Schuster also presented the data in 2015 and 2016. This is the ASH presentation 2016 of the summary in the very high-risk DLBCL subset of the 13 patients enrolled in lymphodepleting chemotherapy. Again, very similar, various regimen. And the overall response they saw was a 52% at 3-month mark. So CR rate about 38%, kind of similar to the data they were seeing with the CLL patients as well. But again, also similarly, no patient in CR relapsed with a median progression-free survival of 5.8 months. What about the other groups? So in order to improve the outcome, so this group at Seattle have adopted a different approach. So the trial that I mentioned before and then was unselected CD3 population, which is probably the most common way of doing it. But they were asking the question, if you define CD4-CD8 ratio, so collect the 1-to-1 ratio of the CD4-CD8 positive cells, is that going to change the outcome of the patients in terms of the increasing efficacy and potentially less toxicity for these patients too? So this is a study that was being conducted at Fred Hutch Cancer Center in Seattle in all patients, ALL, non-Hodgkin lymphoma, CLL patients. This is kind of the basket trial for them, but we'll be concentrating the cohort of non-Hodgkin lymphoma and the CLL patients, which they have presented at various meetings in the last 2 years. They also, the T-cell doses, I should also mention, are different too, which makes it even challenging to compare trial by side too. So T-cell dosing here is about 2 times 10 to the 5 to 2 times 10 to the 7 CAR T-cells per kilogram, just to give you some idea of what cell numbers they were talking about. So again, as I mentioned before, they actually first collect the white blood cells. The previous trials, we mentioned the CD3 is all collected. You just isolate the CD3 positive T cells. Here, they isolate CD4 separately, CD8 separately, and then you transduce the cells separately. You grow them out separately in a 1-to-1 ratio, and then you mix the product together and then infuse it into the patients. And then this one, again, has a 4-1BB as a co-stim domain too. So this is their non-Hodgkin lymphoma data of the 41 patients that presented at ASCO of this past year, 2016. So predominantly DLBCL patients, but they also have some mantle cell lymphoma patients and follicular lymphoma patients as well. These are previously heavily pretreated patients with some of the patients having prior auto or the allo transplant. Lymphodepleting chemotherapy, this group is using predominantly fludarabine and Cytoxan conditioning method. So they used to begin with the Cytoxan. In their data set, they saw they improved the persistence of the T cells and response rates with the flu-cy combination. So they have moved on to use flu-cy combination in their subsequent the patients. So 71% of the patients in this data set have received the flu-cy combination for conditioning chemotherapy. So the response rate, again, the CR rates were about 33%, so similar range is what we have seen in the other study as well. Again, this is kind of the breakdown of the difference. So this is to highlight with Cytoxan alone, and at least in their particular construct and particular T-cell manufacturing process of a CD4-CD8 ratio, the CR rate with the Cytoxan alone was low in 8%, but it was improved when the flu-cy was added with a 50%, although this is a small subset, but in a kind of encouraging response rates nonetheless. So the similar finding is as the previous studies, all CR patients had a very durable outcome. So not everybody stayed in CR, but those patients did much better than the patients obviously who did not achieve CR, including PR patients. So the CR patients appears to achieve a durable outcome, even though the outcome is shorter than the other study that I just showed you at UPenn with about a year follow-up. So based on that study, there is a kind of and there was a single center study. There is an ongoing multi-center study. They named their CAR-T, Jae 17 by Juno Therapeutics with the Transcend NHL 001. The multi-center study enrolling patients with DLBCL with a grade 3B follicular lymphoma and mantle cell lymphoma patients, again using flow cytoconditioning conditioning chemotherapy based on their single center study data with escalating doses of CAR-T cells. This is currently enrolling in many different centers now. They presented their data at ASH. Of the 39 patients have enrolled, 14 patients have been treated, and 12 patients are evaluable for response. So 10 of these patients had a prior transplant, mostly auto transplant, and their response rates in the DLBCL cohort, which was a predominant disease, actually I think all patients might have been DLBCL, is at 9 out of 11, or the 82% of the patients' overall response rate, with a CR rate of 73%. So it's quite encouraging. The response rate, again, is a smaller subset now, so we have to see what the data will look like, but certainly is as good as what the phase 1 single center study have been shown. So what about at MSK? So we actually initially studied our first patient was in 2007, in patient with a CLL. That's how long this has been going on for many, many years. And now we just begin to talk about it in the last 3 to 4 years, but this has been ongoing for several years. And as I mentioned before, that we first started the patient with the treating patients without conditioning chemotherapy. That's actually before we realized that conditioning chemotherapy was an important part of this second generation CAR T cells. So initially a very low response rate that we have published before. Subsequent to that, we went back and realized why the response may be lower in the patients with the CLL, and then concomitantly we were doing the study in ALL at the same time, and we are seeing the discrepancy in the CLL, and other groups have realized that the T cells, and it's just been known way back in the past before, are the T cells, because we're taking the T cells from the patients. If the host T cells are not working properly, they may impact the efficacy of these CAR T cells as well. So the CLL T cells upregulate inhibitory ligands, inducing impairment of T cell immunologic synapses, and that's been kind of shown in several different preclinical studies in the past before using patient samples. And then the CLL cells can induce T cell exhaustion. If they get exhaust fast, then obviously you're not going to get a long-lasting response for these patients too. Perhaps that's the reason that we're not seeing the response that we wanted to see. So, and then there's some data also showing the published in Blood of last year too, that when the ibrutinib is added as one of the other kind of the off-target effect of the ibrutinib or on-target effect of ibrutinib using ITK, that it may impact these T cells to expand further. Meaning they increase the T cell expansion and then the engraftment of these T cells in the patients as well in the in vitro data. So based on this, and we and others have modified the trials, and I believe the UPenn, this was the data that was presented, published from the group at UPenn, have combined the ibrutinib with the CAR-T, CD19 CAR-T cells too. So we have done several patient clinical trials with the ibrutinib is adding at the time of T cell collection, and then the treatment concomitantly to see whether they can improve the outcome of these patients too. So this is in vitro T cell expansion after cell collection while we're growing the T cells in the patients who had a prior ibrutinib exposure. Even though it's a very small subset of the patients, there's a big caveat for that. But the fold expansion by day 7 was higher, although not reaching statistical significance. There's a trend toward the higher fold expansion of the T cells early on. What about the clinical outcome? Again, these are very small subset of the 11 patients that we have treated. This is ibrutinib naive patients, meaning no prior exposure to ibrutinib, and this is exposed patients. The complete response rate was at 27% in our data group. So MRD negative, as controversial as that may be, it was at 18%, and the exposed group was, with the ibrutinib exposure, is about 20%. Subjective response about 80%, and then ibrutinib combined together. And similar to our groups, even though CR patients are relatively small in number, all CR patients continue to remain in remission as well. So we are now doing the trials with the ibrutinib combined with 1928 Zeta. So I should also mention that MSK, we're using CD28 as a co-stim domain as opposed to 2 other studies that we have mentioned before that we're using 41BB. So the one last trial that I'll mention is a KITE trial. This is ZUMA-1, KITE C19. So this is, again, for data that was driven by the NCI group the before. So this is containing CD28, their co-stim domain, using retroviral gene transfer modality. So this is a multi-center phase 2 trial. There's their pivotal phase 2 with 2 cohorts of DLBCL and primary mediated B-cell lymphoma. The eligibility criteria for this study is refractory and progressive disease within transplant. And again, they're using flow cyte conditioning with a cell dose of 2 million CAR T cells per kilogram, kind of similar ranges as the CAR T cells may have been. They have enrolled a lot of patients. This is 111 patients. They presented data at ASH. It's a late-breaking abstract, the 51 patient data. The overall response was a 76% with a 47% CR at 1 month. At 3 months, it was a 33% CR, so there are some drop-off after 1 month. Some of the patients progressed very early on within the first 2 months of the waiting, with a progression-free survival at 3 months about 56% in this high-risk patient population. And I will mention about some of the side effects, and then to mention cytokine release syndrome neurotoxicity, which was seen about 30% of the patients had a grade 3-4 neurotoxicity, and then the encephalitis as well. Grade 5 event in that particular study. So I talked about some of the efficacies, but there are, with this CAR T cell therapy, there are very unique toxicities that's associated with the CD19 CAR T cells, maybe true with the other non-CD19 CAR T cells as well. We have seen with the BCMA targeting CAR T cells too, with the 2 major toxicity that's unique is cytokine release syndrome. That results from the T cell activation and then immune activation as well. So patients will experience a fever, hypotension, respiratory insufficiency, capillary leak syndrome, and then that's correlated with a pro-inflammatory cytokine elevation in serum. And the other unique toxicity where we don't really quite understand the mechanism of why this is happening is the neuro changes, but we do see them in patients with ALL, CLL, and non-Hodgkin lymphoma in various degrees. These patients maybe get delirium and aphasia, dysphagia, word-finding difficulties, but in some of the severe cases, they can get global encephalopathy, obtundation in those patients. These are largely reversible events, although there have been very few non-reversible fatal events in different studies as well. In some cases, the seizure-like activities or the seizures may happen too. So there's a big effort and ongoing to really understand what are some of the patients at high risk of developing neurotoxicities. It's usually the patient with a high tumor burden receiving high T cell doses, but is there other clinical factors or biomarkers? So there's a lot of work needs to be done and is being done to understand that. And just to highlight how the toxicity in the CLL non-Hodgkin lymphoma patients compared to an ALL, this is the data from the Fred Hutch Cancer Center. This is a basket trial that we looked at before. It's an opportunity to look at with the same CAR T cell design, what are the differences. So CRS grading, so 3, 4, 5. There are various definitions, but for the purpose of this talk, meaning the severe degree, were quite similar between those two, although they're slightly more common in ALL, but there's, about 10% of the patients with non-Hodgkin lymphoma had it, about 23% of the CLL patients had at least grade 3 or 4 severe cytokine release syndrome. That usually typically means that hypotensive enough to require vasopressors in ICU. So neurotoxicity rates are also similar, about 25% overall across the board. So that is, these are grade 3 or 4, so usually in a very altered mental status with obtundation. So that is a reversible event, but having said that, it can happen in these patients too, in non-Hodgkin lymphoma patients. And similarly, our toxicity at our group also showed that in CLL patients, the severe grade 3 toxicity was seen in 1 patient, but these are very small group, but I will say that the toxicity profile that we are seeing in the CLL non-Hodgkin lymphoma patients are slightly similar to what we are seeing with an ALL patients too. So the last 3-5 minutes that I have, that I will talk about is that the CLL, just put in the context of CD19 CAR T cells or non-Hodgkin lymphoma, in an ALL patient, we're seeing 70 to 90% CR rate. And I talked about that before, and there's a multi-center phase 2 global trial actually ongoing for phase 2, hopefully lead to an approval of these agents in pediatric and ALL. But in CLL patients, non-Hodgkin lymphoma patients, CR rate, while encouraging and very durable, the rate of the response is low. And then the part of the thing is why that may be the case. And there are many theories, but one of the theories may be the hostile tumor microenvironment. ALL cells are very sensitive to chemotherapy, and you can actually cure ALL, as difficult, aggressive the disease may be, you can actually cure ALL with a combination chemotherapy, which is not the case with a lot of aggressive lymphoma patients or the CLL patients, certainly, although there are subset who can be cured. So there are a lot of tumor microenvironment that we're beginning to understand that may kind of in place both in the bone marrow and the lymph node that may impair the activity of the CAR T cells. Because of that, there's efforts to move in many different ways with MSK that we're also taking an effort of creating armor CAR T cells too. So how can we modify the tumor microenvironment and how can we really increase the potency of the CAR T cells that we are infusing into the patients too. So one of the ways that the new clinical trials is entering is we're opening up later this month is some of the armor CAR T cells. There's one of the ways the 4-1BB ligand expressing CAR T cells. There's another one that's open later this year with the IL-12 secreting CAR T cells that's targeting CD19. So how is the 4-1BB ligand and the CAR T cells really work is that this is our backbone of the second generation CAR T cells, 1928Z. So 4-1BB ligand will actually express in tumor cells and on other, or the target cells to stimulate the T cells that binds to 4-1BB, which is expressed on the T cells. But by expressing 4-1BB ligand on the surface of the T cells, actually, they can actually co-stimulate. You can actually stimulate your own T cells that's modified, but you can also modify the neighboring T cells as well to bystander T cells to increase the potential efficacy in the basically taking advantages of the endogenous immune system to enhance the efficacy. So this was based on the data from the Dr. Sedlin's lab at MSK in the preclinical models, and that's now beginning to move to the phase 1 clinical trials. They're opening this later this month too. We're hoping that this will be the better or the improved efficacy than what we have seen with the second generation CAR T cells too. Another way is the IL-12 secreting CAR T cells. It's again based on the 1928 plus an IL-12. So while IL-12 as a single, as a pro-inflammatory cytokine, but it has an advantage of inhibit the efficacy or overcome the inhibition by regulatory T cells. So T cells can stay active for a longer period of time, and also they modify the NK cell activities too. So we have to see whether what we have seen in the in vitro data will truly be true in clinical, but these are some of the things that we're doing. So where are we now with the CD19 CAR T cells and heme malignancies? So this is a relatively short story compared to the other ones too, but having said that, we've been doing this, there are others, and we and others have been doing this in 2007 as well. So we're now beginning to see some improvement with encouraging data. These armor CAR T cells and newer data with a combination with the immune checkpoint blockade is already ongoing at several centers too. And then hopefully later this year that we get a first approval of CD19 CAR T cells in pediatric ALL that's ongoing by Novartis too. So I kind of hopefully should say that CAR T cells can induce smaller CR, but up to 25 to 50% and 45% of the CR in a very durable outcome without any further therapy after single infusion of the CAR T cells. That's encouraging for these patients, but the lower rates may be due to tumor microenvironment. There are ways that we're thinking of overcoming to improve the efficacy of these CAR T cells as well. Thank you for your attention.
Okay, so the speakers come